So, this little summary table here about the expression of class 1 versus class 2 molecules. So, here's all different kinds of cells. Here's whether they express class 1 or class 2. So, here are the professional antigen presenting cells here. There's also, I haven't talked about this, but there are special cells in the thymus where the T cells grow up. But they also function as professional antigen presenting cells. But broadly speaking, it's B cells, macrophages, and dendritic cells here. And these are the big main guys that present class 2 molecules. Okay. For the class 1 molecules, everybody, okay, red blood cells are non-nucleated, and so they don't express any kind of HC. Okay. But everybody does it, but in particular, also the professional antigen presenting cells express a lot more than other kinds of cells as well. Okay, but for our purposes, everybody made class 1, just a few cells made class 2 with the professional antigen presenting cells. Okay, and here again we've talked about this, but again we have here then the polymorphism of these MHC molecules. You have a very large number of allelic versions of it. Okay. So the class 1 we've talked about here, at 250 known alleles for the B gene protein. This is, I'm sure, different regions and societies and geographical areas and different kinds of pathogens around the world have MHC ones that have evolved to deal with whatever pathogens you find in your particular local area here. So I'm sure we have not exhausted the total number of class 1 antigens that are found here, but we know of some 250 for the B gene, some 70 for the C gene, and 120 for the A gene. Here, for the class 2 molecules, we have the DP gene. Now remember, class 1 molecules have a single big polypeptide chain that makes the entire peptide platform. Okay? Whereas class 2 molecules have 2 genes, A and B, and the two of them, A and B, together make the peptide presenting platform. So we have 2 genes for the class 2 and just 1 gene for class 1, right? So class 2 molecules, we have the so-called DP locus, DQ, and DR. It's a little bit more complicated here. I'm not going to ask you to go into this level of detail here. But we have, for DP, it's A and B, DQ, A and B. For DR, it's A and B, but it could be B1, and then we could also have a B3 or a B4 or a B5. Okay, so some have B1 and then either B3 or B4 or B5. Don't trouble your heads about this. I'm just saying there's a little extra diversity, allelic diversity in the class 2. And when you look at so we have 250 allelic variants of this class 1. Well, so allelic variant means nucleotide changes in the gene, which correspond to amino acid changes in the protein. Okay, and where are these amino acid changes? Well, as you might expect, they're all right in the region where you bind the peptide. Okay? So here are red dots that represent where the amino acids or the different amino acids are found in the different kinds of alleles. So one allele, we've got this guy here. Another allele, we've got this guy. even some alleles, we've got two different ones here. But they're all clustered in the little mountains here and in the platform valley here, just where you're binding. So these mutations affect the different peptide sequences that can be presented or not. And here's the mutations found in the B protein of the class 2 molecule. You can see where they all cluster there. No surprise there. Again, as I've said, each MHC allele or isoform finds a characteristic set of like 10,000 peptides. But they all have the anchor amino acids, only 2 anchor. Well, there's one with just 1 anchor amino acid, but by and large, 2 anchor amino acids. Okay. So where do these allele variants come from? And just broadly speaking, and I'm not going to ask you this on the test either, but there are 2 broad mechanisms that occur, and in our germline cells, when you're making sperm and egg and so forth, somehow we have an increased sort of mutation rate in this particular set of MHC genes, basically. And in some cases, we will have for example, the mother chromosome and the father chromosome, they'll line up next to each other in this region here, and there'll be a recombination event where a little bit of the nucleotide sequence of exon 2 DNA will be popped into the corresponding exon in the dad gene here. Not shown, of course, the dad's yellow exon would be popped up here into the mother's red number. Now, this particular recombinant allele here of the class 1 B gene is found. You find a lot of it in African populations where there's a lot of malaria, so it somehow contributes to malarial resistance, that particular mutant allele of the MHC1B gene. So this is allele 5101, this is allele 3501, and now this recombinant guy is called 5301. Okay. And sometimes it can occur not between homologous chromosomes like this, mother and father, but just as in, say, the father chromosome. Okay, or actually when we made this mother. So here's the mother chromosome. Here is HLA-B, and then downstream from this, HLA-C. This is MHC1A and MHC1. This is MHC1B. MHC1 C gene here. Okay, MHC is the same thing as HLA. So this is the B gene, and this is B1501, and then this DNA can kind of loop around, as you can see here, and now the gene for the MHC1C protein is looped up and it's lined up right next to the B gene version here, and now we get a recombinational event here, and here we have this, and from the zoom we should have put in a little red corresponding part over here. Exchange of DNA there. Okay, and these alleles are given. There's an immunology workshop group, and so there are workshop numbers. There are this workshop numbers that you see to refer to here. And here's a table I want you to memorize for sure. All these different alleles are named as children. That's a bad joke, of course. And you could quit early, because here's, this is not even in your notes, so never mind this, we're done. And I'll have your vote posted by the end of the